So welcome to the first h-ALO webinar, and today we will speak about biosensing for food safety, quality and authenticity. The webinar will be given by Dr. Jeroen Peters from Wageningen University. We will have a session where Dr. Peters will present the topics that have just highlighted, and then we will have a final session of 10 minutes concerning question and answers. In the meantime, while Dr. Peters presents, whether you have questions, you can post them in any case in the chat box so that we can start already collecting the questions that you may have for the final session concerning Q&A. So, Jeroen, if you would like to start sharing your screen, the floor is yours.
Okay, so welcome everyone. So I was asked to prepare a presentation about biosensing in respect to food safety, quality, and authenticity. And this is a part of the h-ALO project where we are a partner in the consortium. And our expertise lays in the field of course, of food safety in the detection of contaminants and also quality factors. So I'll try to explain something more about that in the coming slides. So the mission of our institute is very simple. We want safe and reliable food for everyone. And everyone also actually involves not only food, but also feed, that is the food that's given to animals. So our institute has quite some activities. We are a reference institute. That means we have national and European reference tasks where we have to measure certain contaminants in food and monitor that. The main activity is measuring and detecting food substances. We look at the effects of the substances on humans and animals. We do that, for instance, with an artificial gut, for instance. We look at food fraud and the composition of food. What we do a lot, and especially in our group, is method development. We monitor safe food production. We also have training and consultancy activities, and we also have a 24/7 incident service. So if there is a crisis like we had with fipronil in eggs, then we will be working around the clock to be able to screen all the eggs that were contaminated. So food safety, why is it so important? Annually, 600 million people are affected by contaminated food. And the worst thing of all is from that 600 million people, 420,000 actually were lethal cases, meaning that you die from eating contaminated food. This food can be contaminated with microorganisms, parasites, and chemical substances. Those are the main ones. And you can expect a lot of different symptoms from those contaminations. It ranges from diarrhea to cancer, basically. And of course, lethal, as I said before, you can also actually die from it directly. And therefore, control is crucial. So therefore, we have to monitor foods that we eat, but also the ones that we import from other countries. Since food supply chains is a global network, yeah? So every country imports from food from all over the world or from within the European Union. And that's why the European Union has a general food law regulation where you can find principles, requirements, and procedures laid down in this regulation that we should follow. And most important is the implementation then of control strategies. We have to control that food. And for that, we need detection methods. Those are the basis for everything we do considering food safety. So there is the RAS, the Rapid Alert System for Food and Feed. And this gives an indication on how annually, how many notifications are given, and also how many alerts. And alerts are the most important because the alerts go out, and that means that food has to be taken from the market because it's too dangerous to eat. And if you look at this, there are many classes of food products that can be contaminated, but mainly the highest ranking ones is nuts and seeds. Second, fruits and vegetables. Then you have food supplements and dietetic foods. Poultry, which is a separate class from meat, and also feed and fish products. So those are the main six. This was in 2019, and this can change per year, but still these are always high ranking. Then what do we see the most in these contaminations? And again, the situation of 2019. Most we find pathogenic microorganisms, followed by mycotoxins. Then the composition of food, pesticides, allergens, foreign bodies, which are like, can be metal particles that can be found in food or particles that should not be there. Then contaminations coming from the packaging of food. And then also we have microbial contaminants that are not pathogenic directly, but anyway are a pollution of the food. Now, this is a slide with a lot of examples. I will not go into every detail of it, but just to pick out some. Here you can see where a food originates from. In the case of the first one, it's Gorgonzola, coming from Italy, of course. And then you see the countries which are affected. So it's exported to other countries, and those countries can, in principle, be affected. And listeria, of course, is not a nice contamination to have in your food. And the same holds for pineapples that come from another part of the world, South Africa, and are also sold within Europe and contain a very dangerous pesticide, oxamyl in this case. Yeah, and this raises from the contamination with heavy metals, with mycotoxins, but it can also be, for instance, allergy. Yeah, so if you have this here below, I try to put my mouse. If here below you see paprika powder, which by itself is of course not so dangerous, but if you have an allergy against almonds, well, I don't know even how it ends up in the paprika powder, but it's there. And this can be a risk for people with an allergy against almonds. Okay, then the group that I work in is the expertise group Biosensors and Bioassays. So what we do, we develop, of course, biosensor assays and regular bioassays. So the biosensors is mainly immunoassays, so based on the use of antibodies, and the bioassays where we use cells as indicators for the presence of some compounds. And we look really at a wide range of contaminants. It can be protein-based, like milk quality proteins, but can also be very small molecules like mycotoxins, pesticides, and marine biotoxins. And this list is ever increasing. And the nice thing, what we have as our institute is a really large instrumental analysis department. And the nice thing from that is that it works two ways. So the tests that we develop, we can always control them by our LCMS department or our instrumental analysis department. So we know how good our assay will be. And it was the other way around. When they find interesting contaminations in food and feed, we can use the materials that they find in our validation for the assays that we develop. So if you want to detect food contaminants or authenticity or quality, you have to think, what do you want to do? Are you going to do a routine or research application? Do you want to do that in the lab, or maybe you want to do that on site? So do you need a lab, simple lab, mobile lab, maybe no lab at all, just a kitchen sink? You have to think about it in advance. Are you going to develop a single test like we did for fipronil and X when this contamination was going around? Or do you want to develop a multiplex test like we did for over 100 antibiotics in milk? Then the flexibility, I mean, like I said, 100 antibiotics in milk. Do you want to detect all of them, or do you maybe want a subset of, to detect those? The flexibility, can you determine that on site, for instance? Then, of course, really important, the sensitivity. Do you meet certain maximum levels that are set by the European Union, or are you looking at a zero tolerance? Is it just not allowed to be present? The throughput is very important. How many samples do you need to analyze? And in our lab, mainly we use for that lateral flow devices, or also called strip tests, surface plasma resonance, so that's a biosensing technique, and also Luminex XMAP, which is a very different biosensing techniques, and I will get into these a little bit later. So as I said before, we have quite an instrumental analysis department because we have all those reference tasks, of course, for the government and also for the European Union. And the nice thing of instrumental analysis is that it's 100% proof of contamination. It's an accepted method. And whenever you have to go to court because you accuse someone from selling poisoned food or adding something to food which should not be there, this method will stay also in court. You can really prove that it's exactly contaminated. However, this is a time-consuming method. You need highly skilled personnel. And if you want to detect a lot of analytes in a sample, it will take a lot of time. It's very powerful, however. I mean, in this publication from 2017, you can see that they developed a method where they can detect over 600 contaminants in food, ranging from pesticides to mycotoxins to sweeteners. Of course, this is nice for control instances. However, if you are a food producer, you do not want to detect 600 metabolites in your food, of course, because otherwise every food will be there will be found something, and you want to avoid that. And this has 21 mycotoxins inside this method. However, there are 300 mycotoxins known worldwide. So this is not even complete as a method. But let's switch to biosensors. So this was the golden standard, I would say, the instrumental analysis. But now we go to sensing with biosensors. So we have personal biosensors. What is a biosensor? I think that is something that can sense something that is wrong in case when you look at food safety. So we have our eyes, we have our nose and our mouth. And when we see this, our eyes will tell us this is not good. We should not eat this. However, when we see this, we have to make a decision. Do we eat this, yes or no? And this holds for many things. We can visually see what is there. I mean, you can also taste it. Even the peanuts here on the right bottom, the green ones of sure are contaminated. We will not eat them. But also inside, even if you not see something, our mouth may have some bitter taste sensation. And this can also indicate that probably mycotoxins are present in those peanuts. But what we would like to have, of course, is something really hyper modern where we can actually just look at food and decide if it's good to eat or not. And then we get to the biosensors. So a biosensor, the definition which is most given, is a device which uses a living organism or biological molecules, and then especially enzymes or antibodies, to detect the presence of chemicals. And chemicals can then also be proteins, whatever can be poisoning the food, contaminating the food. Of course, we want some simplicity. This is what everybody wants. We want to make a picture of a tomato, and then we want an answer from our smartphone with an app. What is present in the tomato? Can I eat it? Of course, that would need a miracle process or a miracle app to be able to do that. You get the complete safety information and you can take measures. However, this is something, of course, which is, logically speaking, will never be possible. However, if you look more simple, maybe to pathogens like virus infections in tomatoes, if you would have an app that can do some spectral imaging, you can maybe imagine that this would be possible. But there are other ways. For instance, if we would swap a tomato and we would look at a surface-enhanced Raman spectroscopy, which is also very popular at the moment for detecting pesticides and other contaminants on fruits and other substances. Then we could get a spectral image by this biosensing method. And based on this spectral image, we could say if this contaminant that we are looking for is present or not. And actually, we also work in our laboratory with swabs. We swab cows, and with the swabs we use, we detect antibiotics or antiparasitics. Of course, then we have to make an extract of that tissue that we use. But this is, of course, a possibility. It's also used for detecting, for instance, this is cocaine, this picture. At least I believe it's cocaine, but let's assume it's cocaine. If you see the chip, which is here, there is a small layer. You can swap, actually, or you can put some of the powder on this chip. And then again, you can use this spectral imager to see if it's contaminated with cocaine or not, because of course you want to distinguish from normal powdered sugar. But in this case, you could also debate about if a swab, which you can buy, is not easier, because this is just one analyte, and with the swab it will turn blue, and you know it's cocaine, and then you don't need this imaging analyzer. However, if you go to multiplexing, then detecting more analytes at the same time, then of course the swab is not enough. Then you would need more information. So then it might be convenient again. So if we look at some food fraud quality case, freshness of tuna. Tuna is of course a very expensive fish, but of course it will decay over time. And there are methods to, this is what will happen normally with tuna. So it will start to rot, it will become more dark. However, sometimes you also have tuna, and if you see it, it's really deep red. And the thing is, this can be fresh tuna, but this can also be already decaying or rotting. And why is that possible? So this is tuna that they probably treat with carbon monoxide or some nitrates, and then it becomes red, and it stays red for a really long time, even when it's rotting. So in that case, you can think about color imaging. Yeah? So you can use a color image sensor that can tell you exactly which color it is, and maybe if fraud was used to make it nicer, more red looking, or that it's maybe decaying when it's fresh. And The thing is, if you really would like to detect, of course, if this is rotting or not, you would detect the histamine that is formed during decay. But I will not show examples of that. Then the requirements, of course, the methods that we use, everybody, industry and personal testing, everybody has requirements for that. Of course, it has to be cost effective, it has to be cheaper than cheap. It has to be robust. I remember one time at a Luminex conference, people saying, well, are the machines robust? And he says, yeah, what do you want to do with it? And then he said, we have people that actually can break a sink. So the method that you use, and this may be exaggerating, has to be very robust. It has to be reliable. It means expectations that you have, or that it can answer to EU regulations, for instance. It has to be fast, because time is money, and preferably non-invasive. Avoid preparing, in this case, a tomato soup. So you have to extract your tomato to be able to detect some contaminants. And it has to be preferably portable on site, and then even preferably handheld. And easy to use. If for people that have 2 left hands, it should be actually sample in and answer out. But let's move further into the field of biosensors. So this is actually basically the schedule that you can expect from a biosensor. We have an analyte present. This can be a mycotoxin or a pesticide. It will be picked up by a bioreceptor, which can be an antibody, an enzyme, DNA, or a cell. And then the signal has to be transduced, and then you have to be able to measure that signal. So this is the basic principle for a biosensor. Of course, the most easy biosensor that we all know by now is the COVID strip test. So here an antibody is present in the strip test, which can capture your analytes, and it will get a color formation that we can optically already see with our naked eye. And then we know if we have COVID or not. However, if you go back to this tomato, and then, for instance, for the heavy metal detection, pesticide detection, mycotoxin detection, we would still need to make a tomato soup if we want to have a strip test for the detection of those contaminants. And below, I wrote actually a proposition which was part of my thesis. So because everybody is having rapid on-site methods, develops rapid on-site methods, but the preparation part, so to be able to put your sample in a strip test like this and be able to measure those contaminants, that is a whole different story, and it's often neglected in project proposals. So the test development is important, but I think more important is how do you get your contaminants out of the matrices. To give you an idea about the strip test, this is actually a strip test that is for the detection of the mycotoxin deoxynivalenol. At the left, you can see that's a blank, that's a 0, so no mycotoxin is present, no deoxynivalenol. And then you have 3 ppm and 20 ppm. Now, you can see by naked eye maybe that 3 and 20, that the lines look very different from the blank. However, if you also want to give a concentration to this strip test, then you would also need a machine. Yeah? So you can easily say, I want to have a strip test that I can do in the field. Then, of course, like I just said, the sample extraction is very important. But also, can you really read a concentration? And often you cannot. So to say that a method is portable on site or handheld, yeah, a strip test can be handheld, but if you want really more accurate results, you will always need some equipment for it. Everybody is nowadays using smartphones, and you hear a lot about detection with smartphones, and it becomes more important and more relevant. So what we actually did in our lab with the same tests that were used for these DON contaminations, we made just pictures with a very normal app that you can get, and then you can see that also with a smartphone you could be able to detect a certain concentration of DON. The question is it so important to have a small machine or a smartphone? So this is a discussion you can always have. So within Wageningen University and Research, we have several biosensors. In this case, you can see an octet, an IBIS, and a biocore. And those machines are, let's say, benchtop machines. You cannot really transport them, because when you transport them, they might break down, which is not so optimal. Therefore, we are often involved in European projects where we try to develop handheld or at least transportable biosensors, that they're really stable, you can take them on site and measure your samples on site. And this is just an example of a few of the projects that we were in. Okay, now let's go a little bit into the technology. In this case, you see the technology for the BioCore, which is surface plasmon resonance. So what you have a light source that shines on a metal surface, and on the metal surface here, you can see we have bound biomolecules. And the moment that the biomolecule is binding to the surface, the refraction of light, which happens here, the refraction of light is changed. And the change in this refraction, that is what you actually measure. And that is measured on this chip that you see here on the gold surface. That is where you put your biomolecules. And then you put it in the machine. And this is the detection principle of the machine, the glass prism. And like I said before, if something binds, the breaking index changes, and you can measure that shift in the intensity. That's the BioCore principle. The octet principle, also a machine that we recently are using. It's a bit different. It has tips. You can see the tips, they are going into the biosensor. And on those end of those tips, you can also put a biolayer, which you can see here. A beam of light will travel through this tip. And the moment that something binds to the antibody that is here, your layer will become thicker, and your light that is reflected back to the biosensor will be different. And also here we measure the difference of the reflected light that is traveling back into the machine. So basically, it's all based on let's say, the surface density that you manage to get on a surface, both for BioCore and for the octet. So we have those biosensors. BioCore, well, not this one, is a very modern one. I wish we would have this one, not yet, in the octet. But like I said, we are also involved in European projects, and there we try to minimize, let's say, the biosensor as a volume, as easier to handle, cheaper with the materials. And an example that is the Molecular project that we were involved in, where we tried to develop a photonic sensor that could detect contaminants in milk, but also quality factors in milk. And then you have this very rough setup. You can see we have a separate pump system. Here is the sensor. It has still an external valve. This is the chip with the channels of the chip. And so this is a very rough setup. And this is really a prototype where everything is separate, and you try to get it working, and hopefully then in the future integrate everything in this part that you see here. And people try that all the time. And you can see here on the left, this is also a biosensor, which you can actually already buy at the moment. It only has 2 channels, and the fluidic system, which is also outside here in the Molecular prototype, you can see it's actually still outside of the biosensor. But still, this is a small machine, and this is what people want. They want to be able to go on site, have the strength and the quality of a biosensor, but then be able to do it on a smaller scale on site. Then we get back again to the lateral flow test for COVID-19. I just explained that actually there are antibodies inside this flow test. So this is the COVID particle, the virus. And then when you get infected, your body will make antibodies against this virus. And these antibodies, we can use them actually in those tests to get a rapid screening to make sure that you can tell if you're infected or not. Now, if we look, for instance, at a mycotoxin, in this case, organotoxin A, this by itself, it will not generate an antibody. But if you couple it to a larger protein, and also against mycotoxins, you can develop antibodies. This is a very, very small molecule. So now I will switch to another biosensor that we have in our laboratory. It's based on microspheres, and those microspheres, they have a red and an infrared dye inside of them, and they are paramagnetic, so we can attract them with a magnet. So we can classify a bead by red illumination, by a red LED that you see here. You get a red and a far red image, and overlaying those will tell you which bead we are looking at in this graph. Then we have a reporter signal for when an light is present or absent. It's a green LED. We get orange fluorescent. So the machine will also make a picture of that with a CCD camera. And then it will overlay the bead classification, and it will tell you how much signal is present on this bead. So The biosensor that I showed before, they are label free, so you don't need labels. Actually, here we talk about fluorescence. So in this case, we will need a label. It's a bit of a different principle. We like this technology. We use it a lot in our laboratory because we like the multiplexing capacity of it. Okay, so ochratoxin A, I told you we have an antibody. So what we do, we couple a microsphere with that antibody. And why would you develop a multiplex in the case of mycotoxins? Well, Biomin conducts a survey every year, and they look at the contamination of food and feed in the field. And they test a lot of samples, and what they actually find is that in 87% of the samples, there are 10 or more mycotoxins and metabolites present. Yeah? So if you look for mycotoxins, you will always find more than one mycotoxin, and that's why we would like to make a multiplex out of that. So in that case, it's pretty easy. If you use the microspheres, you just take another microsphere and you add an antibody against the target mycotoxin. And these you can combine all in one single well and do the measurement, and you get 6 answers to 6 mycotoxins. You have to compare this with doing 6, if you know ELISAs, this is like doing 6 ELISAs at the same time, but then in one well. So instead of doing 6 ELISA plates, you do just 1 microsphere plate. Very important, if you put those microspheres together, you have to know that your conjugates, so this is an example of a pesticide assay. So you put your pesticide on the microsphere, and then you have your specific antibodies here, and you do this array testing. And what you want is that your specific pesticide or your specific antibody reacts specifically with your pesticide. And in some cases, you can see that here in this case of the carbaryl microsphere, it actually reacts with carbaryl, but also with the carbamate or the carbofuran antibody. So this is what we call cross interaction, and that is something, if you build a multiplex, that you want to avoid. So that is the difficulty in developing multiplexes. Okay, so for mycotoxins, it's very easy to have antibodies, commercially all available. Now I quickly go to another food contamination problem, and that's marine toxins. So they are produced by harmful algae and bacteria, and they do accumulate in marine animals. And I think one of the best examples actually is this pufferfish that often can contain TTX, which is a marine toxin. And also the effects of these marine toxins can be diarrhea, vomiting, and respiratory paralysis, but it can also be lethal. And what I said before, the TTX in this pufferfish, with one pufferfish, you could kill 30 adult persons. So that means that TTX is really, really toxic. And this is another example of a biosensor, I would say, the mouse biosensor. And this is the most horrible biosensor that I personally know. So what happens here is that, and this used to be the reference method in the European Union. So they make an extract of shellfish, and they inject it in the belly of the mouse. And then the mouse, if it dies, then it means there are marine toxins present. And if it lives, the shellfish is okay and can be eaten. So that's really horrible. And then our colleague in our institute, Anja Gesse, he developed actually an instrumental method for that. So instead of using those mouse, this horrible method, he made an instrumental method where we could detect also marine toxins. Then we thought, okay, instrumental, it needs skilled personnel, it's expensive, takes time. Can we also make a fast assay? So we tried it with cell biosensors, which I'll not go into at the moment. And then we said, can we also make an immunoassay biosensor? Then we started with an antibody against TTX, and that was very successful. We could detect it very easily in mussel, but also in oyster. And we also did that portable on site. That's the advantage of the machine that we use. We can put it in the car and drive everywhere, put it on the bench, and can start measuring. So this was a successful detection of TTX, and we could detect it in all the shellfish. But if you want to have a multiplex, like you can do with the LCMS in this case, you want to add a lot of different marine toxins. However, there are no commercial antibodies available. So here, actually, you can see an example where your biosensor is actually limiting of what you can do. And then you can, of course, say, well, can we raise those antibodies? Because that's normally what happens. If you want to detect a compound in an immunoassay, you have to raise an antibody. But quickly down to the bottom one, you can see that you need to raise those antibodies. Only already the pure material of these marine toxins is really, really, really expensive. I mean, and the last one, if you would do an immunization in mice, you would need 6 million euro to develop an antibody. And of course, nobody will ever try that. So you cannot always have a biosensor where you use antibodies and detect all those marine toxins. Now I will give an example using also this microsphere-based assay of something we developed in the Moloco project, the European project. So that is about beta-casein A1 and A2. Beta-casein is a protein within the milk, and beta-casein A2 is considered a very healthy protein, but I will get into that. So the variation is only present in bovine, so this difference between A1 and A2 is genotype. Other lactating animals are normally pure A2, and that's the result of 1 point mutation. And you can see that here, there was 1 point mutation. So instead of what normally is present in A2 proline, there is a histidine, and this can be cut by an enzyme. And then you get this peptide, which is developed, and this is considered to have adverse effects for your health. So if we would like to detect this difference, we would need an antibody against the A2 epitope and to the A1 epitope. So we found those antibodies. And what we did, we coupled a bead with an A1 protein and an A2 protein, a microsphere, I have to say. And then, of course, we need an antibody against the A1 and an antibody against A2. So what we got from Parmalat, which was a partner in this project, we got milk from cows that were genotyped as A1, A1, A2, and fully A2. 10 unique raw milk samples. We diluted them 1,000 times, and we did it with a buffer. And then we measured them in this multiplex. And then you can see that this method is nicely capable of distinguishing A1 and A2 from each other. So normal commercial milk is often A1, A2. It's like a mixed genotype. And the people that produce A2 milk, of course, they have only cows that produce A2. And it's also nice if you want to monitor offspring, where you have a cross between cows or young cows. You can, with this method, easily distinguish if they produce A1 or A2. This is pretty relevant because A2 milk is, of course, much more expensive. And everything that is more expensive in food, there will always be people trying to be fraudulent and maybe mix it with A1, A2 milk. So this method can clearly distinguish if that happens. So we can detect 1% fraud already in all species, but sheep. We determine the significance with repeats. We can detect 5% fraud also very easily. And it also works for, and that's where the first sentence came from. I didn't explain that yet to you, so that's why now it becomes more clear. So besides that, we can detect the fraud of bovine milk, A1, A2 in A2 milk from bovine. We can also detect the mixing of bovine milk in other animal species. So other animal species are all A2, and bovine is A1, A2. So we could also detect those in those animals listed here below. Now, food safety is very crucial, of course, I just explained, but also how about food guarantee? What is this bridge? I tried to make a bridge to another subject, actually. We all know that worldwide the bee populations and insect populations are declining. And of course, we know that bees, they are attracted to flowers because of the nectar, and they collect the nectar, and by doing that, they pollinate flowers. However, nowadays, in regions in China, for instance, there are not enough insects and not enough bees to pollinate the trees. So what we have are people that actually have as their daily job to pollinate the trees, to make sure that fruits are formed. And so this is, of course, very important. And there was a very wise man already quite some time ago that said, well, if we have no more bees, also men and women will cease to exist. Now, why is that? It can be a few factors. It can be colony management, so beekeepers that are not very serious about their job and they don't take care of their colony, but also environmental factors like loss of foraging habits, shortage of floral flowers, and climate change. Then we have, of course, microbials that can be present and kill the bees, parasites, and pathogens. but of course also pesticides. So just to give you an example of what we developed in considering with bee safety, the Chlorpyrifos is one of the forbidden pesticides in the European Union, not only toxic to humans, very toxic actually, but also very toxic to bees. And there was a fruit grower in Austria that sprayed Chlorpyrifos over his fruit trees. And there were bee colonies very close to these fields, and all the bees, hundreds of thousands of bees, they all died. Yeah. So, and then it's not allowed anymore to produce this substance in the European Union, but globally it's still in production, like in China, India. So it can still be illegally bought and used. To that end, we developed a strip test, a lateral flow test, just like for COVID, that could detect Chlorpyrifos in surface waters. In this case, you can see the development in a cassette. Here we did it in triplicate. We took it out of the cassette, and then you can see clearly from 100 ppb Chlorpyrifos to 0 ppb. So this is the opposite of a COVID test. With a COVID test, a line is developed. In this case, a line disappears. That is always with small components, small targets, because they are too small for a sandwich, which is normally used for proteins, bigger molecules. So here we do a competition principle. So the line disappears instead of that it appears. The nice thing is that also here we have actually a small, I mean, the strip by itself we could already call a biosensor, because we do an optical observation ourselves. We use our eye as the biosensor. But you have also very small handheld machines that can actually be put over the strip, and then you can measure, in this case, the blackness of the strip. And then you can see using that it's actually pretty accurate detection, very stable detection. So using that, you could also actually quantify these strip tests. We used it on surface waters as well. So we collected surface waters throughout Europe in all kinds of parks, rivers, ponds. And then you can see that we can pretty sensitively detect at 2 ppb the Chlorpyrifos in those surface waters. And next we tried it for honey. So we made dilutions of honey that we fortified with Chlorpyrifos. And then you can see that also here we can detect up to 0.22 ppb in honey, which is of course very sensitive. However, this is only for one pesticide, of course. And of course, preferably you would detect multiple pesticides. Now I make a switch to the h-ALO project at this point. So this presentation today originates from the h-ALO project that we are in at the moment. And in the h-ALO project, we also try to build a sensor where actually we can get away from the big lab machines and make everything more simple, more compact, using, for instance, light emitting diodes, organic photodiode detection strategy. And we wanted to do, with this sensor, we want to try also to do localized surface plasma resonance. So basically the same principle as you would see on the BioCore. But we also wanted to try to, because LSPR is label-free detection, but we also want to try plasmonics and enhanced fluorescence. So here actually we are not label-free, but we also want to try to do it with a fluorescent label, see if we can gain something there. And this project is focusing on microbial and chemical contaminants from in the farm to fork food chains. And what do we detect in this project? So it's a unique combination if you want to detect microbial contaminants and also chemical contaminants at the same time. The targets in this project are antiparasitics, insecticides, herbicides, antibiotics, heavy metals, and microorganisms. So it's quite a lot. And the targets that we are looking at, the matrices that we are looking for those contaminants are craft beer, milk, honey, and aquaponics water. Now I'm going back to the bee. So this is something that we actually already developed on the microsphere technology that I just showed you before. So what we call the Bplex. So in this Bplex, we have 8 microspheres that can detect over 28 pesticides. And all those pesticides in this method, they are actually very detrimental to bees. And this was the basis of the h-ALO project, because all these assays are also very relevant for the targets that we have in the h-ALO project. So this was already, let's say, of a high TRL level to integrate into the h-ALO project. And then you can see here a table actually that now we are working on. So besides the B-plex that I just showed what we already had, we actually made the plex much larger, where we have also glyphosate, azoxystrobin, a fungicide, and also the antibiotics. And we added additional pesticide, also with a focus on, for instance, antiparasitics in milk. Yeah, so that's the second group that you see on the slide. That's those. We can now detect all those in milk. We use that for the microsphere technology, and now we are at the point that all these assays are going to be transferred to the h-ALO sensor that we are building in this project. I think for albendazole we already had some preliminary experience on the nanoplasmonic rating of the h-ALO sensor. So this will happen all in the next few months within this project. This already brings me to the end of my presentations. So if you have any questions, please, I will be happy to answer them.
Thank you very much, Jeroen, for this very informative presentation. We already have one question in the chat from Andreas. Regarding microsphere detection, how can multiplexing be achieved? What is the advantage compared to microarrays?
So one of the main advantages for microarrays, because we also work with microarrays, is that you couple those microspheres with your antibodies, and you can store them for over 2 years. They stay in solution. Normally microarrays, they fall dry, and we have often problems in getting to reactivate the chip, let's say. The other thing is with the microspheres is flexibility. So we also develop those assays, for instance, for inspectors, that they go with a machine on site and they can determine there on the spot which microspheres or which targets they are going to analyze. So you don't have a pre-spotted chip. You can decide at the moment what will be your array that you can use. So those are for me the main advantages why we prefer to work with, let's say, the XMAP Luminex technology compared to microarrays.
Thank you. If the other participants have any question, you can also open your microphone and pose it live. Otherwise, you can write it in the chat. So if you have any additional information for our speaker. So I would say that if there are no additional comments, we can close the webinar. I can probably try to unmute you, Andreas, if you would like to speak. Let me see. You should be able to speak.
By ourselves to ask questions. Just maybe for the multiplexing part with the microspheres, is it that you use multiple fluorophores then to label, or are these just different beads already?
So yeah, so the power of this technique is actually in the microsphere themselves. So they contain certain ratios of red dye, and they contain certain ratios of far red dye. And they can mix this in for the 2 dyes, it's in 100 different combinations. And the machine can say which bead it's looking at. So it's illuminated by a red laser or red LED. And then you get a red signal and a far red signal. And then the machine knows which bead it's looking at. And you know which analyte you couple to which bead. So then you can actually use one fluorophore for the detection.
Oh, okay. That's very clever. Yeah.
Yeah, so the beads, I mean, that of course, that's also patented and everything. That is the power. But you can imagine that in hospitals, this technology, they use it for cytokine profiling, for instance. And before, for cytokine profiling, they had to do like 20 to 30 cytokines to be able to, for ELISA. So 30 ELISA plates for profiling a disease. And now And also samples, of course, are very scarce. So now they have to do one measurement where they have all 30 answers in one well. So that is all based on the identity of the microspheres.
Okay.
So now all the participants should have the possibility to unmute themselves. So in case you have any additional question, you are free, of course, to pose it. So if not, we can conclude our webinar. So the first webinar, it's a series, so you will have some more in the future. You will receive information whether you were in agreement with the reception of the newsletter. And in any case, you will find all the information in our project website and the media channels. So thank you very much, Dr. Peters. Thank you very much to all the participants. You will have the chance to have a look again at the webinar once it's published on our channels. So thank you very much and have a nice day.